Hello class, in today's lecture we are going to discuss about analytical method validation. So it is the part of validation, and if we talk about validation, that it is the assurance of the quality of a product or process. So here we are going to discuss about the validation of an analytical procedure. So it is according to ICH guidelines, ICH quality guidelines, which should be maintained by the company. So here we are going to discuss about what is a requirement validation, to know about analytical method validation. So principle is guideline present information on the characteristic to be considered, manufactured to demonstrate analytical procedure is suitable for its intended purpose. So the analytical procedure which is used, it must be according to it must be suitable for the purpose for which it is intended. Validate analytical procedure whether they indicate stability or not. So it must be stable. This is the requirement of the adoption of a process. So validated by R&D before being transferred to the quality control when appropriate. Analytical procedure to be validated: identification test, quantitative test for impurity content, limit test for control of impurities, quantitative test of the active moiety of the drug substance or drug product or other selected components in the drug product, dissolution testing, determination of particle size. So these are the procedures which are to be validated. Why validation? So when should verification or revalidation to be done? Changes in the process for synthetic of the drug substance, changes in the composition of the finished product, changes in the analytical procedure, transfer of methods from one laboratory to another, changes in major piece of equipment or instrument, extent depend on the nature of characteristic and evidence of analyst proficiency. Documentation of validation protocol include procedure and acceptance criteria, report documented results. It will contain justification needed when now non-pharmacopoeial methods are used if pharmacopoeial methods are available. Justification to include data, example comparison with the pharmacopoeial methods, detailed standard test methods. Validation characteristic that should be considered during validation analytical method include specificity, linearity, range, accuracy, precision, detection limit, quantitation limit, and robustness. So accuracy, the degree of agreement of test result with the true value or the closeness of the result obtained by the processor to the true value. How much accurate the results are, it is to be tested. So it is normally established on samples of the material to be examined that have been prepared or to quantitative accuracy. Accuracy should be established according to the specific range of the analytical procedure. Precision analytical procedure expresses the closeness of the agreement between a series of measurements obtained from multiple sampling of the same homogeneous sample under the prescribed condition. So repeatedly we will take the to the sampling and we will check it out. So all the results must be precise. Repeatability, a minimum of 9 determination covering the specified range of the procedure, 3 concentration per 3 replicates each, or a minimum of 6 determinations at 100% of the test concentration. Intermediate precision within laboratory variation, usually on different days, different analysts and different equipment. Right? So precision between laboratory results. Robustness, ability of the procedure to provide analytical result of acceptable accuracy in procedure under a variety of conditions. So when we are going to change the condition, and after that also the accuracy and precision must be maintained in the analytical procedure. So results from separate samples are influenced by change in the operational or environmental conditions. Robustness should be considered during the development phase and should show the reliability of an analysis when deliberate variations are made in method parameters. Factors that can have an effect on robustness when performing UV analysis include stability of the test and standard sample and different lots, and suppliers and temperature. Linearity, it indicates the ability to produce results that are directly proportional to the concentration of the analyte in sample. A series of samples should be prepared in which the analyte concentrations span the claimed range of the procedure. If there is a linear relationship, test results should be evaluated by appropriate statistical method. A minimum of 5 concentrations are used. Range, it is an expression of the lowest and highest level of analyte that have been demonstrated to be determinable for the product. The specified range is normally derived from linearity studies. Specificity, it is the ability to measure unequality, the desire unequally the desired analyte in the presence of components such as excipient impurities that may also be expected to be present. An investigation of specificity should be conducted during the validation of identification test, the determination impurities and assay. LOD, detection limit, limit of detection. It is the smallest quantity of an error that can be detected and not necessarily determined in a quantitative fashion. Approaches, instrumental or non-instrumental, visual evaluation, signal to noise ratio, standard deviation of the response and the slope, standard deviation of the blank calibration curve. LOQ, quantitation limit, limit of quantitation. It is the lowest concentration error in sample that may be determined with acceptable accuracy and precision. Approaches, instrumental or non-instrumental, visual evaluation, signal to noise ratio, standard deviation of the response and the slope, standard deviation of the blank calibration curve. These are the references. Thank you.